Welcome back. In the previous video we saw how the circular DNA in prokaryotes are compacted by formation of loops and supercoiling into a region in the bacterial cell called nucleoid. We'll move on with the compaction in eukaryotic cell, and as the name suggests, eukaryon, U means a true nucleus. It has a true nucleus. And the DNA size also we measured. We had certain calculations done in the previous video. So you know the size of the human genome per cell is around 2.2 meters. So such large DNA, how is it compacted to be fixed into a small sized, very microscopic sized nucleus? So, and what is the need for such compaction? Just imagine a situation like this. You have threads at home, but somehow messed up. See the tangles. Can you work with this? In this situation, it is so difficult to work with. How can you pull out just one strand of the thread to work with? It's just not possible. So we organize it in this manner. And that too, we organize different colors in different roles. So that is the most organized form of keeping it manageable to work more efficiently. Similarly, imagine a tangled up situation of the DNA within the nucleus. The cell won't be able to work efficiently. So, to manage such extremely thin strands of the DNA, it needs a highly organized packaging system as is present in the eukaryotes, so that it can fit such lengthy DNA molecule which measures about 2 meters approximately in length per cell, and that too this DNA is not a continuous strand. So when we put each DNA strand end to end, then it measures around 2 meters. So how is it managed? So this kind of organized packaging is needed so that the lengthy DNA molecule is fit into chromosomes. One DNA compacted into one chromosome. So when I talk about one chromosome, it is a haploid cell. This is one chromosome. This is one pair. That means one paternal and one maternal. And nuclear DNA does not appear as free linear strands because it is organized in this manner. It is highly condensed to be fit inside this nucleus. Now, the dimension of a typical nucleus of a eukaryotic cell is just 10 raised to the power minus 6 meter, a few microns. Whereas the length of the DNA double helix comes out to be so much of meters, 2.2 approximately, far greater than the size of the nucleus which has to hold it. So we'll go through the various levels of packaging. So if you, this is suppose one chromosome being, opened out. You just see the unit. What makes it into that compact structure? The first level of packaging is called nucleosome. The basic subunit of a chromatin is called nucleosome. What is a nucleosome? It is a complex of histone protein and DNA. So, histone proteins make up the core and the DNA is wrapped around it. We'll see in detail in the next frame. How many histone proteins are there? 8 histone proteins, 4 types of histone proteins in the core, multiplied by 2, that means each are in pair. So H2A, H2B, H3 and H4 each into 2. So you can see the arrangement, 2 each inside. So 8 histone in the core. Around 200 base pairs of DNA helix wraps around it. But 200 base pairs is a rough calculation of the total DNA including the linker. What comes around the core histone is normally mentioned as 146 according to Wikipedia. But then the numbers differ in different books. But we'll go with 200 base pairs in all for the examination point of view for the board. So that is the first level of packaging. Then the linker DNA, that is the entry part and the exit part, is sealed by this linker H1 histone, which is not part of the nucleosome, which is making the DNA even tightly packed. The proteins are added to the DNA to make it more compact. One may think when proteins are added, then the DNA may be taking more space to fix in, but that is not the truth. Within the nucleus, these histone proteins provide energy, mainly in the form of electrostatic interaction, to fold the DNA tightly and fix it into the space. So, chromatin can be packaged, the whole chromatin can be packaged into a much smaller volume than the DNA alone, than the length of the DNA alone. Now, the most important part of how is a nucleosome formed? What makes the DNA wrap around the histone octamer? We know that DNA is negatively charged, and that is because of the phosphate group. Histones are positively charged. This is one very important question which normally is asked. Why are histones positively charged? These basic proteins are rich in lysines and arginines, amino acids, lysines and arginines. So these lysine and arginine have their side chains which are positively charged. That makes the protein acquire a positive charge. So this histone octamer, we call it octamer because it is a unit formed by 8 molecules of histone, namely these. You have to remember the names of the histone. And they occur in pairs. So 4 are there into 2. So let us see how they package the DNA into nucleosomes. Since lysine and arginine have positively charged side chains, they will definitely hold on to the DNA. But the uncharged interaction domains will hold each of these 8 histone molecules tightly together. And those positively charged side chains being distributed on the outer surface will interact with the negatively charged DNA backbone. The sugar phosphate backbone there is negative because of phosphate group. So the number of positive charges and the position of amino acids in the side chains are also very crucial for the ability to bind the DNA very tightly onto the histone, because opposite attracts, we know. So that is the first level of packaging. And each such nucleosome, this is just one portion highlighted, each such nucleosome appear as a bead, just like how you see pearls arranged on a necklace. So the whole chromatin appears like that with beads on a string. So we see the nucleosomal histones are wrapped around by DNA, and the DNA that wraps around one octamer is said to be 1 3/4 round on the octamer and then clamped by H1 protein. So such repeating units of nucleosome make up the chromatin, which appear as thread-like colored bodies in the nucleus when stained, when the cell is stained. So we go to the next level of packaging. So this is the previous recap. Genes are part of DNA, codes for a specific protein. This is wrapped around, making nucleosome. Then so many nucleosomes, they appear as beads on a string. So that is what you call chromatin. That means repetitive nucleosome being interlinked by linker DNA, form a complex of DNA and protein. Students, this is just an extra screen I have added because second level of packaging solenoid form is not included in NCERT. But still you should have the basic idea about how these beads are further compacted. So see, 6 such nucleosomes are twisted around like this, and this is where H1 has a role. The linker H1 protein brings them together. This is the top view. This is how you will see in the side view. So this form is called a solenoid form. You can skip this if it is not of your interest, but then we will go to the next packaging level. So we'll talk about the looped structure called chromatin fibers, how the nucleosomes coil and stack together. This kind of compaction involves certain non-histone chromosomal proteins. That is a higher level packaging where these non-histone chromosomal proteins take part. What is non-histone chromosomal protein? See, there are stages in cell when the chromosome has to undergo replication, transcription, etc. This happens in the interphase stage where the strands of the DNA have to be freed out from the histone, core histone. So the histone would move apart, it would release the strands for transcription, replication, etc. But the shape of the chromosome should be still maintained. So non-histone proteins play a very important role. They are also known as scaffold proteins. which play a very important role in maintaining the shape of chromosome even when the histone proteins are absent. So in the interphase stage, the genetic material appears as chromatin fibers. And we know that S phase is where replication of the DNA occurs. So the content has already doubled in the interphase stage, but it is not visible then. It is visible only during cell division, where the more compaction occurs and prophase chromosome appears somewhat like this. Then the next higher level, which is still more condensed, metaphase chromosome appears like an X-shaped if the centromere position is here, and the shapes vary, but most of the chromosomes appear in this shape, with the two sister chromatids being shown very clearly. And this is the result of that replication that has occurred in the S phase, and the duplicated chromosomes are visible only during metaphase stage. And that is because all the chromosomes are aligned in the center of the cell in this stage, in their most condensed form. So it is the highly condensed state and easiest to distinguish and study them. The difference between all these terms from gene to chromatin, chromosome, chromatids, etc. I'll be clearing it in a more simplified manner in the next video. Next is, let us see the regions of chromatin. You will see different regions, namely euchromatin and heterochromatin. Euchromatin region is loosely packed. The nucleosomes are far apart, so they are loosely packed. Whereas heterochromatin, they are densely or tightly packed. The nucleosomes are very close to each other. So when you stain the cell, the euchromatin region appears light stained. Light colored bands are formed. Whereas on staining, this appears very dark, dense or intense staining is seen. Euchromatin is transcriptionally active. That means mRNA can be formed on these DNA strands because they are easy to be separated off from the histone protein. So DNA can be condensed as it is lightly bound onto the histone, so that it becomes accessible for binding by factors responsible for transcription. That means making an mRNA which is finally going to translate into protein. And other proteins needed for replication are also formed in this region. So that is transcriptionally active. Transcriptionally inactive area is the heterochromatin, because the DNA is highly condensed, tightly bound, so not very accessible to the factors responsible for transcription and all. Most of your genome, the significant part, that is around 90 to 92% of human genome is euchromatin, whereas the smaller part of the genome is the heterochromatin. Of course, the DNA is more in this area, so we have most of the genes which are not coding. Found in prokaryotes and eukaryotes. Euchromatin you will find in both, whereas heterochromatin you will not find in the prokaryotes. That shows that heterochromatin has evolved later. So that's it for now. I hope you understood the topic well. If any doubt, you can ask. Till we meet with the next video, keep learning and keep watching. And don't forget to subscribe, please, so that you can get the videos as soon as I put it. Happy learning. Thank you.